genetics, epigenetics, biochemistry and the emotions. Disc 2, Chapter 6.
Okay, so we're going to start off this afternoon looking at functional genomics. So we want to go through this fairly fast because of the time to cover all the contents of the presentation. We know that enzymes are the readout of the gene. Okay, not all genes encode for enzymes, but all enzymes are encoded by genes, because genes encode for amino acids. Okay, so each 3 of the nucleotide bases we'll see encodes for a specific amino acid, and proteins are made of chains of amino acids. So proteins are a direct response to the gene expression, which is why there's all the interest always in proteins in biochemistry and not the cofactors and coenzymes and carbohydrates and things that we put the emphasis on. So we know that enzymes are proteins, but enzymes are results of the gene expression. But just remember that not all genes encode for enzymes. Okay? There's 4,000 or so enzymes catalogued in the database. There's various components to an enzyme, and there are 2 types of enzymes, those that need coenzymes and those that don't. And those that require coenzymes are about 25%, a quarter of all enzymes, such as the oxidoreductase. So those that add a hydrogen, take it away, are oxidation reduction enzymes. So they need a coenzyme. And the thing that does most of this is the things like the activated vitamin Bs. So NAD, which is the active form of vitamin B3, and NADH, which is the reduced form, would tend to just alternate, so that it donates the hydrogen, takes it away, and so on. So that's called oxidation reduction. And there's those that don't require a coenzyme, such as the digestive enzymes. And we know that all the digestive enzymes need zinc, which is a cofactor, a mineral cofactor, but they don't need vitamin Bs, etc., to activate them. Enzymes are proteins that increase the rate of catalyze of chemical reactions. All chemical processes in living cells are carried out by enzymes. And enzymes are usually specific to that particular pathway, or that's what they thought. So oxidase reductase enzymes are those that catalyze oxidation reductase. Then there's transferase, those that transfer a chemical group, such as a methyl transferase, known as those that transfer methyl groups. Hydrolase, which catalyze the hydrolysis of various bonds, known as the adding of water molecules. Lyase, these enzymes cleave bonds by other means, known as they cut bits out or cleave. Isomerize, which means they change the shape of the molecule, but the molecule remains the same. And ligases, which catalyze the joining of 2 molecules with a hydrolysis in between. So there's 4 parts to an enzyme. There's the active part, which is the protein part in there, and then you've got the rest of the string of the enzyme, which is non-active. The coenzyme is that's required to activate and donate something to the enzyme pathway, and those are usually, if we just say activated vitamin Bs, there's a few others which don't fall into that, but generally they're vitamin Bs. And then you've got your cofactors, or your metal ion catalysts. So to us, we put more emphasis on coenzymes and cofactors, the minerals, to activate the enzymes than we do on the genetic aberration or the end result. So to a biochemist and a pharmacist, they would give the end result, the end product. Here, have a pill. So you're low in serotonin. You want a happy pill? Here, have this, okay? Or we'll stop you from breaking it down with a selective serotonin reuptake inhibitor, okay? The orthomolecular approach of Linus Pauling, 30 years ago, would be to increase your tryptophan. Let's give you 5 grams of tryptophan. That'll make you produce some serotonin, It'll force the increase the substrate. What we would say is, no, assuming you're not deficient in tryptophan, but you are deficient in serotonin, let's get you to make your own. Okay? And it may be that you're short of pyridoxal 5-phosphate or zinc or magnesium to open that pathway up and to stimulate its production. Okay? So in fact, we would say the substrate builds up and you get a higher level of substrate because you're not making the end product. So in a person who's sad, got the blues, the depression, they would strengthen to serotonin, let's say, but they would weaken to tryptophan because they got a relatively high amount of it. Okay, so this is how we develop functional biochemistry, is to see which one the person needs. So if you had a build-up of tryptophan, you would weaken to it because it's relatively high. It's relative, okay? And you would strengthen to serotonin because that's what you need. So that weakening with the tryptophan would be negated by, let's say, P5P, pyridoxal 5-phosphate, and or magnesium, zinc, whatever, okay? So we would therefore prescribe the cofactor and the coenzyme rather than just hammering in greater and greater quantities of the substrate. So the common cofactors or metal ions are your tightly metal bound ions such as iron, copper, zinc, manganese, molybdenum, sort of things we saw in various equations this morning. And then there's the loosely bound ones, which are more the big electrolytic compounds, and these are usually in solution, and these would be your sodium, potassium, magnesium, and calcium. So these are what they call electrolytes. Okay, and then lastly, many of them have what's called an allosteric effector. These are things that can change the shape of the active site of the enzyme and allow other things to bond on. So here we see a functional site on the protein. Then we see a regulatory site substance come into here, a modulator molecule. Okay? This then attaches into here, which changes the shape so something else can bond on. Okay? So in other words, they act as like supercharges or something to upregulate the enzyme. These are called allosteric effectors. And the one I always quote here as an allosteric effector is silver. There is no enzyme in the body that is known to be silver dependent. Okay? So silver is not considered to be an essential nutrient. On the other hand, we all know that silver upregulates the immune system by upregulating the myeloperoxidase enzyme, and it does this as an allosteric effector. So what it does is it locks on to the myeloperoxidase, which opens up the functional site and allows something to attach to it to get the immune system working better. So those are called conditionally acceptable, conditionally essential substances. We also know that citric acid is one of these. Citric acid is a well-known allosteric effector, and it upregulates many enzymes and functions within the body, and that's why we add it to all our inert vitamins, and our minerals are citrated. Okay, they're not citrates. Okay, there's a difference in that. So when we use zinc, we use the most soluble forms of zinc, which is the chloride and the sulfate, because chlorides and sulfates are the ones which they register in water. When you look at that, you'll see the anion and the cation. So the cation is the mineral part, the metal bit, and the anion is the carbonate, the phosphate, the sulfate, the chlorides, and so on. And what you find is the most water-soluble forms in water that they measure are chlorides and sulfates. Okay? Carbonates are somewhat soluble, but not always. But we definitely, on muscle testing, we find that the chloride and sulfate is the most ionic or soluble form. And then we add citric acid. Okay, so they are not citrates. That's the difference. Zinc citrate is nowhere near a total form of zinc for everybody. But zinc chloride, sulfate, which is then citrified with citric acid, is. So you can be pretty sure that if a person doesn't strengthen to zinc, then the answer is they don't need zinc. Okay, if it's in the citrated form. Okay, but if you test them with, say, zinc picolinate, zinc citrate, zinc whatever, you can't be sure that they don't need zinc, because they may need an alternative form. One person may strengthen to a sulfate, one to a chloride, and so on. But by making that combination, you're guaranteed you've got a pure soluble dissolved dissolution, as it's called, or dissolved solution of zinc. So the free metallic ions in the water there. And the reason I learned that is simply what they label on water, on mineral water. They don't label things that are not soluble because they form sediment. Okay? They want you to buy this without a lump of chalk in the bottom. Okay? In fact, if you bought a bottle of water with sediment in the bottom, you probably wouldn't buy it again. So they don't measure that. They measure what is here when they've removed all the impurities. So what is in here is ionic. In other words, it's pure solution. And how you tell that is you get a laser, okay, which there's just about some in there, and you put the laser through the water or the solution, and you look. You can see it coming out the other side. All right, see it in my hand. But you cannot see it through the solution. If you see the laser tracking through the solution, it means there's something bigger than an atom in there. Okay, so that means that you've got clumps or colloids. So a colloid solution, you would see the laser track through the solution. Okay, so you know that this is a true ionic solution. So if you do the same thing with all our liquid minerals, not vitamins, liquid minerals, put a laser through, not that you can do that with mirror on glass, but put it through, pour it into a glass, and you'll see that there's no sediment in there. Okay, hopefully. Okay, so silver would be a classic allosteric effector, citric acid is a classic allosteric effector, and there may be lots of other things which can act to boost enzymes up in their function. So here we see the substrate, here we see the end product. So the substrate will weaken, the end product will strengthen. Does that make sense to everybody? Okay, that's what functional biochemistry is all about. And the enzyme we know is an expression of the gene. So providing your gene is being expressed all right, then you should have a normal enzyme. Now we're moving towards what happens if the gene doesn't express itself properly. So that gene expression requires mineral cofactors to attach to the enzyme to activate it. So this is your zinc and your molybdenums and your manganese. And then the coenzyme is an activated vitamin B. An example here is NADH, which donates the H to the pathway and forms NAD on its own. So that NAD then has to obtain another H from somewhere else to recycle. And this is why vitamins like the vitamin Bs, you don't need much. You pee some of this out, but the amount you pee out, most of it you hold in, luckily. So you have to replace that you lose. So you're usually talking for most vitamin Bs of about a few milligrams. Now, if you remember, a paper clip is a gram, which is 1,000 milligrams, so the amount to replace is very small. You know, you're lucky if you see a couple of milligrams. And in some cases, some nutrients, the recommended daily allowance is micrograms. So you wouldn't even see it. It would be a particle of dust, the amount of B12, for instance, that the books tell us we need. But in our experiences, they don't. You usually need far greater quantities for optimal health. Those amounts which are recommended are to prevent known deficiency diseases, not for optimal health. So the person strengthens to the end product because that's what they need. So if we challenge then an enzyme for down regulation, we have a strong muscle weakens to the substrate because you've got a relative excess. The weak muscle then strengthens to the end product, and the weak muscle strengthens to the coenzymes and or these cofactors and or the coenzymes. Right? That may all make sense, all right? Okay. So let's press on through because we want to cover this, the nuts and bolts. You can read the notes all about the enzymes. So the nucleus of the cell is the chemical brain of the cell. Okay? So the nucleus is, if you like, the brain of the cell, or it was thought to be the brain of the cell. The brain depends upon having information. Our brain depends upon information which is put into it, as does the nucleus of the cell. But the brain and your skin are intimately connected to ectodermal tissue embryologically. So the theory is now within the field of epigenetics that the nucleus is like the blueprint of the cell, that when there's damage to the cell, the nucleus releases or expresses the gene for the protein to remake or remodel that damaged tissue. Okay? But the information about the environment comes from the receptors on the cell membranes or the skin. Okay? So the brain, therefore, this is just the plans of the building. So if there was a part of this building collapsed, you would go to the architect and say, show me the plans, now I can remodel. You wouldn't just get the builders in to build, okay? So the latest in the epigenetics is that the nucleus is actually the reproductive organ of the cell and not the brain. The brain is actually the cell membrane, and this is gaining ground. So the cell membrane we know is intimately connected to the brain embryologically, and the brain depends on information that goes into the brain, okay, through the 5 senses. That's how the brain learns. Everything, as we'll see later on, depends upon what you've seen, what you've heard, what you tasted, what you touched, and what you smelt, okay? And then everything you ever thought about or could think about is what you've experienced. You could only have thought about something if you've seen it, heard it, tasted it, watched it on telly, whatever it might be. Okay? And it's the same for the cell. The cell nucleus can't work at all unless it receives information that there's a problem. So it has to receive information to tell that nucleus to produce a particular enzyme or protein to rebuild. Okay? So the receptor is the real brain. And what happens is you have receptors on the cell membrane and we have receptors on the organelles within the cell for molecules to attach to. And there is a cell membrane around the nucleus of the cell. And what happens in these cell receptor sites is they're either on or off. In other words, they open or they close depending upon what is attached there. And what some of them do is allow sodium to go in, and some of them allow calcium to go in, and so on. But the nucleus ones are different. The nucleus allows certain substances to go direct to the nuclear membrane and switch the receptor on or off. Other ones go to the cell receptors, the cell membrane, and then carry that message with secondary molecules, like phosphatidylenosytyl, cyclical AMP, etc., across to the nucleus. So they can't go direct across. But the nucleus is somewhat different. This is the nuclear membrane here. This is the gene, and the gene then it makes a template of messenger RNA, which travels out to the ribosome here, which builds the protein. It's transcribed here. But to switch the gene on, you need information, and that information has to come from the receptor side. And they thought that these hormones, such as steroid hormones, such as thyroxine, the very big regulating hormones, can actually go through the nuclear membrane and directly attach to the hormone receptor site on the gene. Okay, so that's what made them different. The latest sort of more epigenetic information is that this is probably not chemical but mechanical, and that we know that proteins are strings of amino acids, and that some amino acids have positive charges and some have negative and some are neutral, which means they have a configuration. So if we look at my arm, we go down here, up, across, and let's say that, all right? Now, if something attaches to here, or on the receptor side here, on the protein, it'll change the charge. So let's say something attaches here, and it changes the positive, and you lose the positive, it'll straighten out, okay? So if something attaches here, it may straighten out. It changes its shape. And that changing when something attaches to the receptor site has, by the series of the protein, connects into the gene here and actually mechanically turns it. So the protein, there's a protein that links here on the receptor side, called an effector protein, or effector receptor, into the gene. So what happens is something attaches like a hormone or something that acts on that, called a ligand, and it changes it, and it turns. Mechanically, the protein turns and operates the gene. Now the gene, we put all the emphasis on the gene structure 50 years ago was on the DNA, on the nucleic acids in there. And they know that when they melted cells down, there was protein in there, nuclear cells, and DNA. And all the emphasis went to the DNA. But now they realize, why was the protein there in the first place? And the protein is there as a sleeve to protect the gene. Otherwise it would get damaged from oxidation and etc. So what happens is it's like a sleeve. So the gene is not exposed. Okay? So along comes somebody, a receptor on the receptor site, and attaches to the receptor site and moves the protein, which changes its configuration, and it pulls it mechanically. Isn't that amazing? And it opens it up, and then it's red, and then it goes back in. Okay? Now, what you want to do is make sure that that stays like that. What you don't want is more exposed, because what they know is that each gene refers to a particular protein structure. But if you expose a little bit more, you get a different protein. Okay? You get something completely different. Well, we know now with the Human Genome Project that there's roughly, and it's always roughly, nobody seems to come up with a firm figure here, 25,000 genes. Now that sounds a lot, doesn't it? But they were hoping for 150,000 genes, because there's 150,000 identified proteins in the body. So there must be 155,000 or 150,000 genes. Okay, this was the Human Genome Project. The Human Genome Project was not for humanity. It was a business venture. Okay, it was a massive, massive corporate venture financed by big money to make pharmaceutical drugs and license each one, patent each one for a particular gene. So pharmaceutical companies could make a lot of money because they'd had the actual world patents on them. And lo and behold, as the results came in over the last 10 years, they discovered they're in 25. Which means now, A, the Human Genome Project was defunct because it wasn't one protein, one gene for one protein. It meant that some genes could encode for maybe hundreds of proteins. And what it depends upon is how much is exposed. It's as simple as that. So if something attaches to here and alters the configuration, it'll encode for a protein. But if something else attaches to there, it could encode for another protein. Okay? So that code could be carcinogenic. Okay? And if that substance that attaches here is, say, a pseudo or xenoestrogen, we could have all hell break loose, because you'd have here something which is activating the production of estrogens when we don't want it. Okay? And they tend to stay locked onto the receptor site much longer than real estrogen, because real estrogen would dissolve within minutes, but pseudoestrogens stay on the receptor site for a much longer time. So what we want to do is to keep that hidden, if you like, and used and open and shut when we need to. Now, round these proteins are called histones. And on the histone there's a little tail which keeps it wrapped round, like a piece of sellotape locked on there. And we know that a lack of methyl groups slackens that tail and allows more of the gene to be exposed. And this is why methylation, as I saw this morning, is the critical thing. Now we know acetylation stimulates the removal of exposure of the gene, and methylation keeps it closed. Okay, it keeps the protein round much stronger. So methylation in this case is really, really important, and particularly for blue people. Okay, when we look down at that homocysteine S-adenosylmethionine chart, we can see the blue person there, you know, and we can see the coronary artery type person, the person who's high in homocysteine and the person who's low in homocysteine. So when we fit it all together and look at the body types, you'll say, before I've even put the slide on, about what a red person needs nutritionally and what a blue person needs. And you'll say, now I understand why Chris has said that every single person who weakens to red needs the following things. And you'll find the same thing. It'll be consistent 100%. Providing, of course, the person is red, green or blue truly in the beginning. Because we're not always who we think we are we? Sometimes we're disguised as somebody else. You know, we did this in the update a few weeks ago, that, you know, practically, you know, half the group were not who they thought they were. We had greens becoming reds, we had reds becoming blues, and all sorts of things. And really interesting, and we had combinations of people who weakened to red and blue, and so on. And then they ended up as being green. So we've got to get to the core of who you really are. Okay? And when we discover the core of who you really are, what your constitution and body type is, then you'll say, yes, that explains why I need the following nutrients, and why I need to follow the following diet, and I need to take the following oils, and I need to avoid the following heavy metals and things. So that's basically how it works. So let's not get too involved now with basically what I've been talking about. Sorry, we're whizzing on. You can read this. Okay, now, DNA and RNA are composed of nucleotides, which are derived from substances called purines or pyrimidines. The purines bases are adenine and guanine, and the pyrimidines are known as cytosine, thymine, and uracil. So these are pyrimidines. Uracil is normally in RNA and replaces thymine, which is in DNA. So DNA would be adenine, guanine, cytosine, and or thymine. Okay? So we'll see that those are the four bases, but there's no thymine in RNA, but there's uracil instead. That's the dream ticket. Okay? Now these are often abbreviated to A, G, C, U, and T. Okay? So sometimes that's how they're referred to. Nucleosides are when the base has a sugar added to it, which is either ribose for RNA or deoxyribose, which is deoxynucleic acid for DNA. Nucleotides are when there's a phosphate, a monophosphate added. So there we've got AMP, or cyclical is AMP, ADP, and so on. So here's the bases: adenine, cytosine, guanine, thymine, uracil, and then adenosine, cytidine, etc. as when the sugar's added. And when you add the phosphate group, we can get AMP, CMP, etc. So when you add all those together, these are called nucleic acids, and those are known as RNA or DNA. So DNA is composed basically of phosphorylated, sugared bases. Now it's the bases that sometimes can get in the wrong position. Don't worry about the sugars, don't worry about the phosphate groups. It's the bases which are the important things, because if the bases mismatch, the gene can't be read properly. And this is called a single nucleotide polymorphism, or otherwise known as a mutation. Okay? Something's in the wrong position. So instead of that coding for the production of tyrosine in the protein molecule, it encodes for phenylalanine. So that's going to be disastrous, or it may not matter too much. You know, it depends on the enzyme that it's or protein it's encoding for. So when we come to test for a nucleotide, single nucleotide polymorphism, all we do is put on the bases on the patient. So we put A, C, G, T, and U. And if the person weakens to one of those, you know there's a problem. Okay? And then you find the other one that negates it. So the secret code, which I discovered 10 years ago now, is there's a meaning to this. So if, for instance, G weakens and T strengthens, it means that G has gone into the position of T. Okay? And that encodes for a greater need for vitamin B6, or more specifically, pyridoxal 5-phosphate. So each code encodes, each mutation encodes for a coenzyme. So the possibilities are 4, those could get into the wrong position. There's 5 nucleotides, okay, altogether. So we can get DNA mutations, we can get RNA mutations. So those are the 4 bases that are, with everybody, common to everybody. Every living creature has the same bases. So 4 possibles going into a 5th position. 4 5s are 20. Okay? So there's 20 possibilities between that. So permutations of combinations of defects, and there are 20 coenzymes. Okay? So every one encodes for the greater need for a coenzyme. There's 20 coenzymes. These are all listed for you. Better still, they're on the back of the laminate here. So the other side of the laminate we'll deal with a little later on, but this side of the laminate really gives you the DNA and the RNA mutations. So that's basically doing the same. So here we see the string of DNA. So here we see A, or adenine, T, C, G, and so on. So it's a long ladder. And this is one side of the ladder. Okay? So DNA, we know, is a double ladder with rungs. So this is one side. And you see this oxygen here? This is where we get oxidative reactions. So free radicals or oxidative reactions will lead to free radical oxidative damage to the RNA on these oxygen molecules here. So this, if they're exposed to electrons, it really ruins the side of the ladder. Now, when you have damage to the side of the ladder, you have to repair it. Okay? So something has to come along. So let's say this one is damaged. Something has to come along and cut this out and pop it back in again. Okay, a new one. Okay? This is damage to the side of the ladder. And providing one side of the ladder is damaged, we can replace it. If both sides of the ladder are damaged, we can't. The ladder collapses. So this is more serious. So damage to the DNA is damage to your blueprint. Partial damage that goes on in every cell in your body, you know, thousands and thousands of times, as we'll see, can be repaired, providing both sides don't get damaged. Now, things like superoxide, peroxides, myeloperoxides will do damage on one side. But the big boys, the hydroxyl radical and peroxynitrate, can damage both sides of the DNA ladder, and then you've had it. Okay, as far as the damage, you can't replace that. And that really is the aging process, isn't it? When you can't replace your DNA, you know, your cells are dying out. So we want to preserve that, again, the importance of the right nutrients. So here we see the spiral, and what we know is that the bases will pair together. So if we damage T here, and we've got to cut it out, we'll know always to put a T back in that position, because T only bonds normally with A. Okay, so C will bond with G, G with T, G with C, A with T, and T with A. Okay, so the magic is you get one nucleotide put in, and only one will fit into the lock normally. Okay, so this is the sequence. So when we split it in cell dividing, it creates two strings, so the ladder is pulled apart, but it always knows which ones to put the new molecule on, okay, the new RNA on. So let's have a look. Here we see the code. Let's take the simplest one up here, TTT. That's thymine, thymine, thymine. Now the DNA sentence for the protein is a string of amino acids, because proteins are made of strings of amino acids, and therefore there is a string of nucleotide bases. So the genetic code is three things which encode, let's say, TTT for phenylalanine, followed by, let's say, CCT for proline, CAT for histidine, and so on. All right, so it's a sequence of three. It's always three things. There's no full stops between the sentence, and every sentence begins with methionine. Okay, methionine is the starter for every genetic code. Okay, and then when the code is finished, there's a full stop, and the full stop is TAA or TAG, TGA, TGG. Okay, sorry, that one, full stop. Okay, so there's always a full stop and there's always a start. So methionine, the code for methionine is like the at the beginning of a sentence, or the capital letter. Okay, but there's no spaces between the words. It's one, and that's what encodes for the string of protein. Now, say we've got TAC, okay, which encodes for tyrosine, the amino acid tyrosine. Okay. Now, because of an injury to the cell, to the nucleus, because of a toxin or possibly a deficiency in the right nutrient, we've got an abnormality. We've got one of them goes in the wrong place. So say this C here, which encodes for tyrosine, an A goes into the position instead. So this code here now reads TAA, which encodes for stop. So that protein is stopped at a certain sequence along its chain before it's ready to stop. Okay? That's called a mutation. Now, a mutation may be something which is really serious, and it can affect the person's life all the way through, or it may only affect it partially and decrease the function of that enzyme or that protein, or it may have no effect at all if it's in a non-coding part of the protein where it's not really of any great value anyway. Okay? So it can be disastrous or any phase in between there. But many of our patients do have polymorphisms. Probably we've all got polymorphisms to some extent going on the whole time, but we are able to overcome these by the right nutritional intervention. Now, the right nutritional intervention may be that you need a bit more B6 to upregulate those enzymes. But there are some people, and maybe it's only 5% of the population, who cannot convert sufficiently pyridoxine into pyridoxal 5-phosphate, the active form. Okay? They will obviously convert some, but not enough. And that gene that encodes to convert pyridoxine into pyridoxal 5-phosphate may have been damaged because it's got a mutational single polymorphism, nucleotide polymorphism in its coding. Okay? So that's the code itself. So we know that we form templates. Nucleotides themselves, these building blocks, we can eat, we can create, and we can repair old ones. Okay? So these are the three possibilities. Now, we can make new bases from the raw materials. Now, these raw materials are basically amino acids, aspartate here, glycine here. This is making purines. We need glutamine. Okay? But most important to us, most people are not deficient in the amino acids, is this molecule here and this molecule here, which are forms of folic acid. Okay? 2 molecules of folic acid are needed for every new strand of purine. Okay? That's a lot when we're producing millions and millions every hour. Okay? So that means we need a lot of folic acid. So when a cell is multiplying, like in growth, you know, particularly in the tummy or childhood, you need a lot of folic acid, or rather the mum needs a lot of folic acid as the baby's in there. We know that a deficiency of folic acid will not lead to the correct sequence in the offspring or the daughter cells. Okay? And we know that folic acid deficiency is the cause of neural tube defects. It's probably the cause of a lot of other things. They're now beginning to realize that cleft palates are B12 deficiency and so on. So deficiencies in the mother can cause genetic defects in abnormalities in the cells. Now we see the same thing come back later on in life when we find that we've got a mutation, and one of the common mutations here is U into the T position, which is in other words an RNA nucleotide in a T position in the DNA, and that sequence of U into T encodes usually for methylene tetrahydrofolate, which is the main thing people need with breast cancer. Okay, so we can begin to say, well, cancer, yeah, there could be that there's an involvement with a folate deficiency there. And we know that folic acid is important for methylation and so on. So you begin to build a picture up. So we can make it from those bases, but most important, to make new DNA or nucleotide bases, we need folic acid. So the first thing with any restoration of gene formation is folic acid. We can ingest these from foods. All foods contain the same bases. The only trouble is we don't have the enzymes very well to do this. We need deoxyribonucleases, which will chop up the nucleotide, the DNA, into the base substances for us to reuse. But there is evidence that the body can do this to a small extent, and it has been done by injecting radioactive isotopes of these, and it can be shown that it can go through the skin. So it's possible that some nucleotides could be transported across the skin transdermally. But lastly, and most important to us, is we can repair damaged DNA. So when the DNA is damaged and we've got to cut or cleave out that side of the ladder and replace it with a new one, we have to activate an enzyme called DNA polymerase. Okay? So this is like the cement that puts it back. And DNA polymerase and RNA polymerase, in case there's a mutation there, is a zinc-dependent enzyme. So the basis of all work with polymorphisms and genetics is folic acid, or the various derivatives, and zinc. And strangely, you actually need zinc to activate folic acid. To get folic acid to become the activated forms, like the methylene, methyl, methanyl, the three different forms, tetrahydrobiopterin, all these, all need zinc. So now you know that zinc and folic acid are really, really important. And most things which are made with folic acid are the leafy green vegetables, aren't they? Leafy green vegetables. The greener the vegetable, the more the folic acid. And these are the things that your mummy told you must eat up your greens if you're going to grow. Okay? In other words, you're going to multiply your cells in growth, and those are the things that the kids don't like. They'd rather get their folic acid synthetically from Cocoa Pops, which is probably where most of them do get it from, in fact, because most of them don't eat green vegetables. Okay? And most people don't like green vegetables because now they can taste the nicer insides, because the green ones are a bit bitter, aren't they? But strangely, the green ones also contain zinc. Okay? So the lack of green vegetables is really quite alarming, that people cut green vegetables out of the diet. Now, they also don't cook them properly. Now, if you cook green vegetables, folic acid and zinc go out in the water. So if you boil them up in the water or you steam, it'll go down there. You've got to drink the fluid. Okay? So the real value of vegetables is actually in the water in most cases, not in the vegetable itself. The vegetable will give you the fibre, but you've got to drink the water. So you've got to make a gravy or a soup from that. Okay? So that's very important, how to cook vegetables. So DNA polymerase and RNA are zinc dependent. Okay, so when they are deficient in zinc, deficient in folic acid, we may get something in the wrong position. And that is called a single nucleotide polymorphism. Bruce Ames says, in his estimation, the well-known biochemist, there's 25,000 to 100,000 oxidative hits. That means free radical hits to the DNA and the fatty membrane in every cell every day. Wow. Free radicals are not something because you eat a packet of potato crisps. Oh, I've been exposed to free radicals. Right? Free radicals are natural reactions of oxidative reactions in the body. How did he come to that conclusion? This figure is attained by measuring the quantity of oxidized, deoxidized guanosine, in other words, the damage to the DNA, in the urine per day, and dividing it by the total number of cells in the body. So how many cells are there in the body? 50 billion. Or is it trillion? Billion. What? It's billion, yes. Well, it doesn't matter. Someone must have. But some people got more and some people got less. And that's why there's the variety. Now, if you then eat the wrong foods and set up an inflammatory reaction, you've got a lot of toxic metals in you, you've got an infection, you've got allergy. You can increase this dramatically and destroy your DNA much quicker, with a bit of trying. So as long as you have a cabbage leaf, when you eat your potato chips or and things, it might be right. So the same cofactors work with RNA polymerase. So therefore we need an adequate pool of nucleotides, in other words the folic acid, and we need zinc to activate it. So the best forms of zinc are what I told you, the chloride, sulfate, fully ionized in an aqueous solution,